I want to tell you two stories today, but the most important thing I want to tell you about is what I think most of us working on Down syndrome think is the most exciting thing to come along perhaps in our entire careers, and that is a clinical trial to improve cognition in people with Down syndrome. This is something that even 5-10 years ago, most of us did not think we would see in our lifetimes, and now we believe that there is very good reason to think that we have found a way forward to do this. And for those of you who are maybe at earlier points of your career, this is the promise of what might be done with translational research, so starting out with mice to actually get to people. So the problem I want to talk about is Down syndrome, which results from inheritance of 3 copies of chromosome 21 instead of the normal 2 copies. Trisomy 21 is present in every cell from prior to conception onward. So every function that's regulated by genes, which would be most functions, from conception onwards is influenced by the presence of that extra chromosome. Every developmental process, every functional aspect of life in the adult organism. Chromosome 21 contains between 300 and 500 genes, depending on what your definition of a gene is these days. And so from a reductionist viewpoint, it's an extraordinarily complex problem to think about what each of these genes is doing at all those steps all the way through. And a key for getting at this has been not to try to understand this issue one gene and one aspect of development at a time, but rather to look at bigger phenotypes. So if we start with what we really know about Down syndrome, we can see it right here. This is a karyotype from an individual with Down syndrome, and you see that there are 3 copies of chromosome 21. So we know that an individual who inherits that extra chromosome will have a subset of a very long list of clinical features that are more common in people with Down syndrome than in the population at large. There are only a handful of things that actually occur in just about everybody who has trisomy 21. So variability actually is one of the first things we run into in looking at this population. So in order to look at these complex outcomes all the way through and not knowing which genes from this large set to begin with, an animal model becomes very important for that, because we aren't just asking how does this gene interact with that gene, we're asking how these cells interact with each other, how this tissue that's been formed from those cells affects other tissues, how the organ systems coming out of that are interacting, and so forth. And so we need an end phenotype that is as complex as the situation we're looking at. We need animal models for that. So as Len mentioned, some of the early work in my lab was involved in mapping genes from human 21 into their orthologs or counterparts in mouse chromosomes. And what we discovered after a number of years of work in my lab and others was that there are 3 clusters, a large cluster at the distal end of mouse chromosome 16, a very small one on 17, and a somewhat larger one on mouse chromosome 10, that contain all the mouse orthologs of human chromosome 21 genes. And within those 3 clusters, the genes are in the same order that they are on human chromosome 21. So even long before we had sequence and could line everything up, we had a good idea that there was a very good genetic correspondence in these regions, that there was reason to think that we could get correlations for trisomies between these very different organisms. Okay, and a very big step towards this process was made by Muriel Davison. Muriel Davison is shown here with one of her coarser tools, but in the laboratory she also had quite a number of very fine tools that she used to actually create trisomy in the mice. And what Muriel did, for those of you who are aficionados of mouse genetics, she wrapped male mice up in lead foil, all except the most interesting parts. She irradiated them, and radiation can cause breaks in chromosomes in spermatogonia, which will reheal to cause chromosome translocations. And she and Cecilia Schmidt then did a truly heroic job of screening thousands of progeny from these crosses to look for new translocations. And based on work from my lab, she knew that translocations involving mouse chromosome 16 could be of interest. So when she found one of those, she sent that mouse to me. We used a now extinct mapping procedure to actually map the breakpoints in all of these translocations that she created. And we found that her 65th mouse, the T65DN translocation, this extra chromosome that she had seen in this mouse, we showed that it corresponded to genes from human chromosome 21. And this, sort of mundane as it sounds now, actually provided us a pretty important breakthrough in work in mouse models for Down syndrome. Up until this time, as a new gene was cloned on human chromosome 21, for those who were graduate students, we didn't do that with Google in those days. You actually had to go in the lab, and it took several years per gene. And so when we got a gene, it was a big deal. And someone would make a transgenic mouse out of that, run whatever test they could think of to run, and say, aha, this mouse has Down syndrome. This is the gene on chromosome 21 that causes Down syndrome. And that actually sounded just as silly then as it does now. But with the presence of a mouse that had first an extra chromosome showing that there's an extra centromere and extra telomeres that has to be segregated, and secondly, having orthologs for what we thought was probably about half the genes on human 21, we had a good genetic basis for now asking the right question. And the right question is, can a mouse have Down syndrome? Before you can start using an animal model, you better make sure what you're talking about on both ends. Is this really going to represent what happens? And of course, to ask about representation, we looked at phenotypes. And we looked at a variety of phenotypes that were well characterized in Down syndrome at that time. So the first one we looked at is short stature. This is not very specific, but it's very easy to measure. We measured the growth of the mice, both as mass and also as long bone length. And indeed, the trisomic mice were smaller than their euploid littermates. And I will use euploid throughout here. That's the genetic term from the Greek for true chromosome number. So trisomic mice and euploid mice. The second thing we looked at was the face. One of these handful of features that's common to people with Down syndrome is the very characteristic facial appearance. And a lot of the appearance of the face in Down syndrome is a reflection of the underlying midface skeleton and mandibular skeleton in people with Down syndrome. So what I've shown you here is a cartoon of a mouse skull and a human skull. For those who haven't had biology for a while, the mouse is on top. And the bones are all color-coded here to show that there's a one-to-one correspondence in those bones despite the 80 to 100 million years of evolution that separate these two species. So when speciation occurs, nature doesn't throw out the recipe for this very complex set of instructions that you need to take a small group of cells and grow them into a large mass of cells that differentiate into these very specialized and tightly fitting bones here. Rather, that original genetic program is modified so that you can get a different shape as the outcome, but that the underlying instructions and the cell populations and the differentiation events that get you there are conserved between those species. So if we have a process whose genetic regulation is genetically conserved, and we perturb that process in an analogous way in mice to what happens in a person who inherits 3 copies of chromosome 21, the question was, can we disturb the development of the craniofacial skeleton in the same way? And I had the very good fortune to have a longtime collaborator and friend, Joan Richmeyer, who was then at Hopkins. She's now at Penn State. Joan is a anthropologist, and she works in morphometry, developing methods for looking at the shapes of complex objects. And Joan and Subhash Lele had developed a program that makes a mathematical model of a complex form and does it in such a way that 2 groups of forms can be compared to each other and analyzed statistically. And statistically, is a very important word in that sentence. There were many morphological programs available that could say, aha, here's a shape, there's a shape, everywhere a shape, they're different shapes, but they couldn't come down to what scientists want to know and say, are those shapes randomly different, or is there really something that sets this one shape apart from the other? So the algorithm behind this, you might guess, is rather complex. It makes an n-dimensional model with I don't know how many calculations, but when Joan flipped the switch on her computer, all of East Baltimore kind of browned out for about a week when she ran the calculations. It's very complicated. And what I'm showing you here is a very simplified two-dimensional version of these n-dimensional models and the comparisons between them. But it proves the point here. The vectors in purple are smaller in the skulls of trisomic mice than of euploid mice. And you can see that all these bones up here, in the midface especially, are smaller in the craniofacial skeleton of the trisomic individuals. And in fact, this corresponds to the flattened maxilla and the other bones in the midface in people with Down syndrome. The vectors in yellow are not different between trisomic mice and their euploid litter mates. Those corresponding vectors show very little difference between people with 2 or 3 copies of chromosome 21. And the vectors in dotted lines are relatively broader in the trisomic skulls than in euploid. So what we have here is a short, broad skull. That's the classic definition of brachycephaly, which is what we see in Down syndrome. And I think the icing to me on this was this vector that's smaller right here. Those of you who know very many people with Down syndrome have probably seen at least a few of them who have a noticeably flat back of the head. And that's because of the development of a bone called the occiput. And this occiput is also flattened in the TS65DN mice. Similarly, the mandible is quite small in mice as it is in humans. Mandibles develop rather differently in these two species, but we know what the developmental fields are, and those points can be compared meaningfully. They're affected in a corresponding way in mice and in people. So this is a pretty rigorous analysis, and it showed us, frankly, more correspondence than I thought we might get. So the model was beginning to look good to us. So, and we now know that there are differences in the palate also, but mouse palate and human palate are quite different developmentally, so we'll let that one go. But what we now call intellectual disability of the Down syndrome type, which wasn't really very well characterized at the time, that was a big challenge. We didn't really know how far we could go. I don't mean to offend the autism community, but I have a hard time understanding some tests of mouse behavior and trying to correspond those to rather subtle human behaviors. And when we looked with what we knew about intellectual ability in mice at the time, this posed similar problems. But what was evident in mouse testing was that we could use tests that were affected by functional or structural problems in different regions of the brain and get some idea about outcomes based on what we saw in these kind of tests. And we did a number of them. I'm just going to tell you about one today, which is called the Morris water maze. It is literally a water maze. It's a tank about 6 feet around. It's filled with dilute latex paint so that it's opaque. There's a platform hidden underneath the surface of the paint. And the mice are trained to find the platform. Mice don't really like to swim very much, but they're quite good swimmers. And after a few days of training, they learn that there's a platform that will let them get out of the paint and not have to swim. And after training them, we then remove the flag that we used on the platform originally, and they now have to navigate to the platform based on markers around the walls of the room that they are able to discriminate from each other. So regardless of where the mouse goes in the tank, it has to remember that the platform is between the heart and the cross and not between the sun and the moon and so forth. And so we time it, we look at the latency to get to the platform when the mice is put in the tank at various different positions. And the trisomic mice are very bad at this task. So this is a euploid mouse, and the number of seconds it takes. You can see that they get there very quickly. The trisomic mice don't do very well at all. There isn't a huge learning curve here. At the end of these hidden platform trials, we call them, we remove the platform altogether and look at how much time the mouse spends in the correct quadrant. And what we find is that the controls have learned very well. They spend most of their time over there. There is learning in the trisomic mice, but not nearly as much as in the euploid mice. So this is a task that, in which the trisomic mice are quite impaired. And this was an interesting thing to find because it had already been linked to function of a specific part of the brain called the hippocampus. And this observation in these mice actually led to 2 lines of research that came back together recently as the basis of this clinical trial I'm talking about. Oh, I need to acknowledge the mouse. There's the mouse. Okay, so the first line of research that came out of this was in people. Lin Adele, who's currently at University of Arizona, he was in Colorado at the time, looked at these results and said, gee, we ought to know more about the function of the hippocampus in people who have Down syndrome. And he actually developed a Morris water maze for children. It didn't involve a swimming pool, so Lin never went to jail for that, as far as I know. It did involve a perfectly symmetrical room with 4 doors, and there were 12 pie plates on the floor in that room, and there was a treat under one of the plates, and each of the walls was marked differently. And so the child had to remember how to get to the plate that had the treat based on the markers on the walls. And just as in the trisomic mouse study, children with Down syndrome did very poorly on that test compared to their age-matched controls. And so this is an indication that hippocampus might be an especially affected region of the brain in Down syndrome as well. And so we now do that actually with a joystick and a laptop. It's a whole lot more convenient than building a room everywhere you go to do the test. But it is the same test of visuospatial integration of markers in order to navigate to a specific spot. So that led Lin and one of his then graduate students, now an assistant professor at also at University of Arizona, Jamie Edgen, to begin thinking about a battery of tests that would be specific and precise for Down syndrome. So an intellectual disability can occur if somebody inherits 3 copies of chromosome 21, if they inherit a mutant copy of FMRP, or if they get hit in the head with the brick. But all of these are causing that problem for very different reasons. And they're affecting different aspects of brain function to different degrees. That was the logic behind this test for Jamie and Lin. So they began to develop a battery of tests that would be especially sensitive for Down syndrome. And that's where I got involved along with Stephanie Sherman. Jamie and Lin looked at 3 regions: cerebellum, which I'll come back to, prefrontal cortex, which is a place that there was information that there were problems with working memory and so forth in Down syndrome, and hippocampus, as I said, which was a target for this test. And they were interested in finding the best tests to look at a broad range of people who had Down syndrome. And so to do this, they had to test a lot of people and decide which tests worked well, which tests didn't work so well. And it's difficult to recruit a large population at one site. It was going rather slowly. So Stephanie Sherman at Emory and I had a network of sites around the country where we were running a project to look at congenital heart disease. Down syndrome is a sensitizing condition for that, which increases the signal-to-noise ratio. And for geneticists, this allows us more power in those studies. So we went to Lin and Jamie and said, gee, if this test is any good, it's got to work more places than just in your laboratory. So why don't we put this into place in our sites around the country? Because they are already at clinical centers. There are people there who are very good at testing in children. And in addition, if we ever are going to have a clinical trial in Down syndrome, it's going to look pretty good to have the most sensitive test available at sites that can already run it and are already recruiting people with Down syndrome. And they said, yeah, that sounds good. So we put it together. And one of the sites now, I just point out, happens to be near home here. And in fact, it turned out that a validated test battery was assembled. It's called the Arizona Cognitive Test Battery. And it's now, although not by name, by practice, a part of the evaluation in the Roche clinical trial that I'm getting to here slowly but surely. And so that's been published, and you can read about the behavioral aspects of that, which most of you will understand much better than I anyway. But what it represents is a sensitive battery of tests that are precisely chosen for people with Down syndrome. Okay, so I said there were two pathways that came out of this original observation of hippocampal issues in the trisomic mouse. The second one is in the laboratory, and it's in the neuroscience area. Zygmunt Galdzicki at the Uniformed Services University in Bethesda is an electrophysiologist. So he measured electrical currents in the hippocampus of trisomic mice. And he found that there was a deficit in LTP, long-term potentiation, which is an electrophysiological process measuring a change in conductance in the main pathways in the hippocampus, and it's correlated with learning and memory. It's thought to be a physiological basis for learning. And when he looked at the TS65DN mice, he found that indeed long-term potentiation was compromised in the trisomic mice compared to their euploid littermates, very consistent with what was seen in that Morris-Water maze test. And this is extended by Zygmunt and also by Bill Mobley's group down the road in San Diego now, who showed that the difference in this long-term potentiation was correlated with and probably fundamentally caused by an imbalance between excitatory and inhibitory inputs into the hippocampus. So to make these complex electrical circuits that are going to be the basis for memory, obviously that's a process that has a lot of fine-tuning, a lot of different inputs to it. And they're both inhibitory and excitatory inputs. In the trisomic mice, the inhibitory input was way out of balance with the excitatory input. And they showed that in these slice cultures, in culture, measuring these electrical currents, if they added a drug that inhibited the neurotransmission in the inhibitory neurons, they could restore that balance, and when they did that, they restored long-term potentiation, the conductance of channels through this pathway. And so that was a very important physiological finding because it put somewhat of a basis onto why we were seeing hippocampal deficits that were pronounced in Down syndrome. So that was taken the next step, as usual, by a very bright graduate student, Fabian Fernandez, who was in Craig Garner's lab at Stanford. And Fabian said, gee, if it works in a dish, maybe it'll work in a mouse. And what Fabian did was to test an inhibitor of these inhibitory neurons, they're GABA-A neurons, and tested it in mice, and he did the following experiment. So this is a test called novel object recognition. It's another test that's been strongly correlated with hippocampal function. And so what Fabian did was to take a bunch of mice, trisomic mice and euploid, habituate them to the test cage, so they weren't excited about getting into that. He taught them to drink chocolate milk. That's about as difficult as teaching your 5-year-old to drink chocolate milk. Mice really like chocolate milk. And then for 2 weeks, he gave them chocolate milk, but half of them got chocolate milk with the drug in it, and half of them just got chocolate milk. So apparently this drug doesn't taste bad in chocolate milk. That was the first conclusion. And he then did this novel object test. So to just put this very quickly, if you put a mouse in a familiar environment that has 2 new objects in it, it will explore those objects, and it will spend, on average, about the same amount of time exploring each of the new objects. And if you wait then some period of time, an hour, a day, a week, whatever your experiment calls for, and put it back in the cage, same cage, but now you've replaced one of those objects, it will spend more time exploring the new object than the other object. Mice are innately curious, and this has been seen in many, many different strains of mice. So it's about 70%, 30% for in this case, a euploid mouse. And the same thing is true if you actually move one of the original objects. So instead of being like this one is in a new position. And once again, using visuospatial integration, if the mouse recognizes that, it will spend more time investigating that object. So when Fabian looked at this, he plotted this out as a discrimination index here. This is what the euploid mice did in terms of their ability to discriminate between the new object and the old. And you can see that the trisomic mice, the mice that, the trisomic mice are very much impaired. So euploid mouse, trisomic mouse, these got milk. The euploid mice do fine at discriminating, but the TS65DN mice aren't able to discriminate here. The mice that got PTZ, there was no difference between the euploid mice and the trisomic mice. So the normalization of long-term potentiation seen in a dish was reflected here in the actual behavior of those mice in a test that depends on hippocampal function. So that's okay. But one of the more compelling aspects of what they did was, after they got the mice to do all the tests, their reward was that they put their brain in a dish and did the LTP measurements. So this is just a measure of a parameter called EPSP of long-term potentiation, and after a certain kind of stimulus, the ability of that network of neurons to conduct in a euploid mouse will be greatly enhanced, and the decay is very slow here. It takes a long time. That's long-term potentiation. Compared to the trisomic mice, you can see that there is quite a difference between them and the euploids. And here are the trisomic mice that got the chocolate milk with the drug in it. And you can see a significant difference here. Now that looks pretty impressive, but what's more impressive is that this actually was done 3 months after the mouse got their last bit of that drug. Okay, so that drug is actually an active suppressor of a channel on the inhibitory neurons, and it is having that drug bound to that receptor that lowers their activity that should be causing this behavior. But that drug's gone. It's long gone. And yet there is a retention in this long-term potentiation effect several months later. If you look at the electrophysiological data immediately after this, the mice that got PTZ will track right on top of the wild-type mice. So there were actually several important things about this. One was the translation from the dish into the mouse. The second one was this unexplained phenomenon, which to me, if I was a neuroscientist, this is what I would be working on. This is whatever this memory is that's going on in these neural networks, in conductivity in these neural networks. That's a very interesting property. But the third one is many people who have a child with Down syndrome get the information or the misinformation when the child is born. You have a very narrow window to do anything about this. A year, 2 years, the brain will be pretty much what it's going to be. You're all over. This experiment was done in young adult mice. So 2 weeks of treatment with this drug in young adult mice was able to produce in the animal a predicted result from the mini results that had been done in the laboratory. And that was in adult mice. So there were many, many aspects of this study that were extremely exciting to the community of Down syndrome researchers. So it still takes a serendipitous element in any big discovery, and this clinical trial is no exception. In 2008, Michael Harpold, who is the Chief Scientific Officer of Down Syndrome Research and Treatment Foundation, went to the neuroscience meeting in Chicago, and he ran into a friend of his, Stefan Navet, who works at Roche. And Stefan was working on a new compound they had that was an antagonist of GABA-A alpha-5 receptors. So actually these inhibitory receptors we're talking about here, this was a drug that turns down those receptors. And Stefan said, yeah, we have this drug. It turns out it has a lot of really good pharmacological properties, and we're trying to figure out exactly what we should use it for. And Michael said, when does the Down syndrome trial start? And Stefan said, in essence, what's Down syndrome? Nobody had thought about that. And they had a beer and a long conversation. And 2 months later, there were half a dozen of us in Basel for an extraordinarily intense 12-hour meeting to discuss every scrap of data relevant to this from the get-go with the investigators at Roche, including the fact that we not only had a very strong scientific result here, but we had the most sensitive detector for change in intellectual ability that had been developed for Down syndrome, and we were already employing it at sites that were custom-made for doing a clinical trial. And Roche looked at all those pieces, and at the end of the day, they wrote the check. And so that trial is actually going on now. We're in phase 1. They had a lot of safety data in what's term of art is normal healthy volunteers. You and I could spoon this stuff on our cereal, actually. But we emphasized to them the fact that there is some literature on the potential for increased seizure activity in Down syndrome, and they agreed with us that it really is worth doing a phase 1 trial. As part of that phase 1 safety trial, they are incorporating most of the tests that comprise the Arizona Cognitive Test Battery as a secondary measure there. So we're just about to go to phase 3 in that trial, the third drug level of that. That should be done. I think the plan now is for Christmas sometime. We hope that it will be possible to get that data summarized and the phase 2 application written and through the FDA in a year, and then we'll see the efficacy trials begin. So this has just transformed the Down syndrome research community. My early grants came back with reviews that said, very good conceptual ideas, good technical approach. It's not worth working on Down syndrome because there's nothing you can do about it. That literally was an opinion from an NIH study section. So we've come a long way since then. And I wish I knew who wrote it so I could rub it. Okay, so the mouse clinical trial results. The genetic models of trisomy 21 show a number of features with a direct correspondence to Down syndrome. I haven't been able to tell you today about cancer resistance, which we've used the models to look at, about congenital heart disease, about a number of features of Down syndrome that are recapitulated with surprising degree of precision in this mouse model. So we think genetically, when we create a parallel or an analogous genetic perturbation in mice, we see comparable outcomes as we do in people. Hippocampal function is especially affected in Down syndrome and the mouse models. The excessive inhibitory input is affecting LTP, and it can be treated both in a dish and in an animal by drugs that antagonize the GABAergic receptors. So the big question, and I think an especially relevant question for the Mind Institute, is will these, I think there's a very good chance that these drugs are going to affect the biology of people the way they're affecting the biology of mice. What I don't think we can really do readily is to say, here's how the mouse behaved, so now we know exactly how the person is going to behave. We don't know what the timetable for adaptive changes might be. We don't know how those might be expressed. We're extremely pleased to be working with Lin in a language battery because we think that one of the things that people are likely to respond with first, if they have improved capacity to do so, is communication. So I think the language battery might prove to be an especially sensitive early indicator of positive changes in the trial. We'll have to see how that goes. Okay, so I want to take a few more minutes here and tell you about what might be a direction for some future research in this area, and that has to do with another part of the brain which used to be thought to be kind of a lump at the back that keeps you from falling over, but we're learning does a lot more, and that's the cerebellum. And this work from a number of people in my lab. This is Laura Baxter. I believe that this is her completed thesis, which is why she has that big smile on her face. Otherwise, people in my lab are always scowling and working very hard, but I allow them to smile on the day they finish their dissertation. So Laura was interested when we first got this mouse model for Down syndrome, and we're trying to figure out which phenotypes we ought to see, look at for parallels between mice and people. We settled early on the cerebellum. So the cerebellum is this part of the brain back here in a midline sagittal MRI section. And you can see that in someone with Down syndrome, it's noticeably smaller. And in fact, on average, it's about 70% the size in someone with Down syndrome as it is in somebody with 2 copies of chromosome 21. So it's a really significant reduction in size. And so the first thing we did was to determine whether this volume reduction was also seen in the TS65DN mouse model. And we did high-resolution 3D MRIs, which now you probably have a facility right on campus to do. We had to actually hijack the protein NMR machine and get somebody to build us a special loop for that and everything. But at any rate, we found that the cerebellum indeed was smaller in the trisomic mice than it was in their euploid littermates. So this looked like it might again be a parallel phenotype between trisomic mice and trisomic people. So Laura went to work on characterizing this histologically, once again midline sagittal sections, and she measured various parameters of size. First she showed that these midline sections at the vermis of the cerebellum, that the reduction in cross-sectional area that we found in TS65 was a good surrogate for the reduction in volume overall. And in fact, it seemed to be pretty uniform. We did these measurements at the midline and out in the periphery and got the same degree of reduction. She looked at the thickness of this dark staining layer, the internal granule layer. I'll say more about that in a minute. The lighter staining molecular layer, at the overall cross-sectional area. You can't see in here, but there's a single line of Purkinje cells, actually a layer of Purkinje cells. And in all of those parameters, the TS65DN mouse was reduced relative to the euploid mice. And Laura then asked an important question. She said, well, the volume occupied by the granule cell bodies that make up this dark IGL layer, that's significantly reduced, but are the cells just packed in there tighter, or what's going on there? And she looked at the density of granule cell neurons in euploid and trisomic mice, and you can see these dark cell bodies. There was a significant reduction in density in the trisomic mice. Okay. So those granule cell neurons actually represent half the neurons in a mouse's brain. And as a matter of fact, half the neurons in your brain are the granule cell neurons in your cerebellum. So there's a lot of them. Maybe they're doing something besides keeping you from falling over and leaning too much on the podium while you give a talk. So Laura found this difference in density, and we looked in the literature. Nobody had ever actually looked for this in Down syndrome before. So the Juan Troncoso in our neuropathology department at Hopkins was able to find us age-matched specimens of brain cerebellums from people with 2 copies in black or 3 copies of chromosome 21 in white. And Laura found that there was a significant reduction in granule cell density at all ages all the way through. So a highly significant reduction. So at this point, we had just used the mouse model, the putative mouse model, to predict a phenotype nobody had ever looked for in humans before. And when you can do that, you get a little more excited about the model. And that led Laura to ask a really good question. She said, if we look back during development, can we find a point before there's a divergence in the development of the trisomic and the euploid cerebellum? Because that would be a very important developmental point to know. It may or may not have its roots farther back, but we certainly should know about that. So she did a really extensive study. I knew there would be graduate students in the audience today, so I made this font so small that you can't possibly read it because you'd probably just give up in despair at what you have to do to get a dissertation. But anyway, she did a lot of analysis here and measured every parameter that she could come up with between the euploid and the trisomic cerebellum. And basically, she found that for anything she could measure, the trisomic mice were smaller by postnatal day 6 days after birth. And they remained that way throughout life, pretty much. Okay. But at the day of birth, she didn't see a difference in the parameters she could measure. And now I want to point out a structural difference here. You see this dark IGL I've been talking about here. At 3.5 weeks, the cerebellum is pretty much completely formed. But at the day of birth, there is no internal dark staining layer there. There is no internal granule layer. Rather, there's a dark layer of high density of cells on the surface of that cerebellum. And what those are precursor cells to all these granule cell neurons that are going to come to be half the neurons in your brain. They're packed in there at very, very high density. And during the first 2 to 3 weeks after birth, they divide like crazy. And as they divide, they migrate inward. And that you can see that internal granule layer starting to form here as this darker layer of precursors divides, moves in, and differentiates to eventually give you this nice IGL here. Okay. So when Laura looked at these cerebella here on the day of birth, postnatal day 0, she saw that the cross-sectional area was the same and the thickness of that layer of precursor cells was the same. But she was also able to count the number of those cells that was dividing, and she found that there were fewer dividing in the trisomic mice. Unfortunately, the way the histology was done, we couldn't really determine a baseline. Were there fewer dividing cells because there were just fewer cells there? They're really packed in here very tight. But there is a way to answer that question, and Randall Roper came to my lab as a postdoctoral fellow and used an approach called stereology to get a very precise estimate of the number of precursor cells. And what Randall found was that on the day of birth, there was the same number of precursor cells in the trisomic and the euploid mice. No significant difference, but the number that were dividing was significantly lower in the trisomic mice. And so, not surprisingly, 6 days later, there was a very big deficit in that precursor cell number in the trisomies. And of course, then there were far fewer cells that were dividing as well. Okay, so this detected not just the time at which development diverged here, but it also showed the cell types that were affected and the granule cell precursors and the process that was affected, this generation of precursors immediately after birth. And there's Randall. He's smiling too. I don't know where all these pictures came from. Okay. So here we actually ran into a bit of luck and actually intersected with another area of science. It's rather rare in Down syndrome, unfortunately. It was known that the mitogen, the growth factor that makes those precursor cells divide, is the growth factor sonic hedgehog. That is the primary and clearly the most important factor for getting those cells to divide. So we looked at sonic hedgehog in several ways to see if we could see whether there was an effect in the trisomic mice. I'm not going to go through all that data now, but I want to show you this graph that was done by another student in the lab, Nidhi Saran. What Nidhi did was to isolate those precursor cells away from the rest of the cerebellum. She put them into culture, and she added increasing amounts of sonic hedgehog growth factor, and she measured the amount of cell division that occurred. That should have been CPM times 10 to the third, I guess, for tritiated thymidine incorporation. And what she found two important things. With the euploid cells, as others had shown before, the more sonic hedgehog you add, the more cell division you get. In the trisomic cells, they responded less at every single concentration of sonic hedgehog. Now, those are 95% pure cultures. So either there's a granule cell interaction, or we think more likely, there's a cell autonomous response deficit to the mitogenic effects of sonic hedgehog. And those of you who've done any developmental biology know that sonic hedgehog is a growth factor that's used over and over and over again in many, many aspects of development. And in fact, we wrote a review recently speculating on whether an attenuated response to sonic hedgehog might underlie a lot of different phenotypes in Down syndrome. So that was the first important thing, was that they responded less at every concentration. The second important thing was they responded. So, and they weren't that far off. At 10 nanomolar of purified sonic hedgehog, they responded about like euploid mice did or granule cells did at 3 or 4 nanomolar. That's not that big a hop. And we thought maybe if we could show those mice a little bit more sonic hedgehog right after birth, when they were not dividing as much as they should be, maybe we could have a positive outcome on the development of the cerebellum. We, since this is recorded, I'm not going to tell you the whole story, but we spent a long time and a lot of money looking for a way to do this and to get sonic hedgehog into the brain. Sonic hedgehog, you may know, is a dually lipidated molecule. It has a cholesterol moiety on it. It's heavily palmitoylated. It's not a molecule that's really easy to just inject into somewhere. So fortunately, we had Phil Beachy, still at Hopkins then. He's now down the road at Stanford. And Phil had shown the ability of a small molecule, potential drug, which he called SAG for sonic agonist, to replicate the activity of sonic hedgehog in every biological assay that they used. So Phil gave us some of this SAG, and we injected it into mice the day they were born. And we asked what happened to the development of the cerebellum 1 week later at P6. Now, I've actually shown you part of this data in a different form before. I showed you that in terms of the number of granule cell precursors, the trisomic mice have significantly fewer than the euploid mice did. But the trisomic mice that got a single injection of this SAG the day they were born, a week later, they had the same number of granule cell precursors as the euploid mice did. And not only that, but when we looked at the number that were dividing, that number, of course, was lower in the untreated than in the euploids. But again, there was no significant difference between that and euploid in the mice that got the SAG. So that implied that we should have a beneficial effect of this stimulation going on farther out. So we've looked in other brain regions for morphological changes. It's obviously very difficult to say nothing changes anywhere. But the places that we thought we might see a change in the hippocampus and especially the dentate gyrus, there was no effect of this. We didn't see an obvious stimulation of cell division by SAG in a BRDU labeling experiment. None of those says that there couldn't be some important replication there. But whatever the effect is, it is long-lasting, and we do know that there is one long-lasting effect of that injection of SAG the day of birth, and that is it normalizes the structure of the cerebellum and the architecture of the cerebellum. So this supports a sort of burgeoning area of research into the role of cerebellum in learning and especially procedural memory, and we think might be an important aspect of looking towards therapy, cognitive therapy in Down syndrome in the future. So I just want to be sure to leave you with the message that basic research has really changed the game for people with Down syndrome. This Down syndrome clinical trial is transformative. We've completely turned the corner in how we're approaching Down syndrome and what the goal of this research is. And the basis for that started with a mouse in a big Morris water maze. Okay, so there are a lot of people here that I have tried to mention from my lab as we went along. The Cognition Project with principals here, and we now have a number of additional sites for that. And I didn't show you all of the analysis from this or the clinical trial, and I would be remiss if I didn't also thank the mice. So thank you very much for your attention.
When you were showing the slide with the box or the square and the triangle, and they had the 50-50 chance of, because they like doing different things, they're curious, which showed that they had, their hippocampus was normal in the euploid mice. So were you trying to say that when you had a square and the triangle for the trisomic mice, that they actually still did not recognize that they had seen that object before, and they always recognized it as new, which proved that there was a deficiency in their hippocampus?
Yeah, so I hope I didn't put too much motivation behind what I said they did, but the trisomic mice did not discriminate between the new object and the old object, whereas their euploid littermates spent much more time exploring the new object.
Okay, and then I just had another quick question. SAG, what is that? I'm sorry, I missed it.
It's a small molecule, a potential drug that crosses the gut and the placenta and the blood-brain barrier, and it stimulates the sonic hedgehog pathway. That's a somewhat complicated pathway. Sonic binds to a receptor called patched, which is an inhibitor of a molecule called smoothen. When sonic binds to patched, it lifts that inhibition, smoothen becomes activated, and starts a cascade of events within the cell, notably with the GLE transcription factors, to affect its changes. There were a couple negatives and a couple positives in there. SAG binds directly to smoothen and activates that pathway.
So basically, it inhibits the neurotransmitter so everything doesn't become excitable.
This is not neurotransmission now, this is cell biology. So this is a pathway that in the SAG and sonic experiments, that growth factor stimulates those cells to divide through a pathway that we think we know something about. SAG short circuits that, but it turns on that same pathway.
I'm not a mouse biologist, but I do know that postnatal day 0 on the mouse is not birth in the human. So could you just talk a little bit about what you think, particularly the experiments that had to do with the timing of development and how that relates back to the human situation and what that might say in terms of, clinical
relevance
interventions
to
down
people.
the line?
Yep. So at birth, the cerebellum in the mouse is probably at a stage that in people corresponds to about 8 months gestation or so. That's a little bit of a tough call to make. In mice, that proliferation that I talked about takes place over the first 3.5 weeks of life or so. In people, that takes place over the first 1 to 2 years of life. So there's still a lot of that same process that occurs in human beings after birth. But it does begin earlier while you're still in gestation in human beings.
So I have 2 questions. So the first one, you mentioned there is an imbalance between excitatory and inhibitory inhibition in the Down syndrome, but you only mentioned the inhibitory synaptic transmission. Can you combine another strategy to increase the excitatory neurotransmission?
Yeah, that's a very good question, and there's a lot of research going on into that. It turns out that the GABA receptors in particular have been a huge target for the pharmaceutical industry for a very long time. We have a very wide range of drugs already available. So this one affects the GABA-A neurons. There's now evidence that you can have an analogous kind of effect targeting GABA-B with known drugs. In some cases, many are already FDA-approved drugs. So the focus has been there, but I think that restoring that balance by increasing excitatory input, which would bring both levels back closer to what you see in euploid, is clearly an area that should be investigated. And the second question?
So, the second question is,
thanks for not asking both at once.
So people now, to reprogram patient fibroblasts into induced pluripotent stem cells and then further differentiate them into neural cells and model human disease in the dish. So you are modeling disease using an animal model. So do you have any comment on the new technology of modeling human disease?
Yeah, we're very interested in iPS cells. We have a project that I hope is going to get funded to make iPS cells from an individual who has mosaic Down syndrome. So some of their cells are trisomic and some of their cells aren't, and otherwise they're completely isogenic. So I think we can do some, get around some of the genetic questions of using unrelated cell lines by doing that. There's a lot of potential for that, as recognized by the Nobel Committee yesterday, for example.
The majority of Down syndrome patients end up developing Alzheimer's disease later on in life. Is there any implications for the use of SAG or other molecules from mouse research in Alzheimer research?
Yeah, I'm glad you mentioned that. Essentially, everyone with trisomy 21 develops the neurohistopathology, the plaques and tangles of Alzheimer's disease. 60% or so will undergo a marked cognitive decline. Those studies may not be as well age-matched as they should. Some of the difference from 100% that would be predicted might be due to earlier death, but there's probably also a different course. There's not a role that I know of for sonic in that pathology at the moment. And the origin of the deposition of that pathology is almost certainly related to the fact that you have an extra copy of the amyloid precursor gene and one of the critical enzymes that cleaves it into the pathological C-terminal fragments off of that. That process is somewhat different in mice. Mice lack the cleavage site for base 2 that forms the amyloid that's deposited in plaques, so the trisomic mice don't show that. You can make plaques in mice if you put in a human APP gene, especially one carrying familial mutations that enhance that process. But I think the relationship between Alzheimer's disease in people with Down syndrome and without Down syndrome has been largely ignored by the Alzheimer research community. And in fact, 2 weeks ago, there was a meeting in Chicago at the Alzheimer Association to see if maybe we really ought to have a little more meeting of the minds between those areas.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.